Welcome to this retelling of Auxin biology, where recent discoveries challenge decades-old models and reveal a far more dynamic signaling network. The slide shows the auxin molecule branching into a complex web of pathways, hinting that auxin's actions are diverse, integrated, and responsive across time and space, rather than following a single, linear mechanism. Auxin truly acts as the master architect of plant form, coordinating processes like gravitropism, phototropism, phyllotaxis, lateral root initiation, and regeneration. The diagrams illustrate how directional auxin transport creates local maxima and gradients. And the text reminds us that this mobile signal reprograms development from embryogenesis to wound healing, explaining its outsized influence on plant life. Plants tightly control auxin availability through localized synthesis, reversible conjugation, and irreversible degradation. With enzymes such as TAA1, TR, YUC, GH3, and DAO shaping, where auxin is active. At the same time, polar transport mechanisms create directional flow. Apoplastic pH and PN family transporters work together to trap and then export IAA, producing the cell-to-cell currents that drive patterning and growth. For many years, the nuclear pathway reigned as the central dogma of auxin action. Auxin acts like a molecular glue stabilizing TIR1, AFB, AOX, IAA interactions, triggering AOX, IA, ubiquitination, and proteasomal degradation, which frees ARF transcription factors to activate Auxin-responsive genes. This elegant mechanism explained transcriptional responses and even served as a blueprint for other hormone signaling systems. But cracks appeared in that canonical view when researchers documented very rapid physiological changes that transcription alone cannot explain. Root growth can be inhibited within minutes of auxin application. Phosphorylation of hundreds of proteins occurs in a couple of minutes, and fast ion fluxes like cytosolic Ca2+ transients precede gene expression, forcing us to ask how a protein degradation model can act this fast. A major revolution came with the discovery that TIR1, AFB proteins also have enzymatic activities. They can produce cyclic nucleotides. The auxin-induced receptor complex, therefore, triggers three distinct outputs at once: canonical AUX, IAA ubiquitination, plus the production of CAMP from ATP and CGMP from GTP, adding new, rapid signaling layers to the familiar degradation pathway. These findings imply a bifurcated signaling architecture with two complementary timescales. The slow arm is a transcriptional CAMP-dependent signal required for canonical gene activation near ARF complexes, while the fast arm is an ion flux pathway tied to CGMP and channels like CNGC14, generating rapid Ca2+ increases, membrane depolarization, and immediate physiological responses prior to transcription. Another revolution revived a cell surface role for auxin perception. ABP1 and ABP1-like proteins act in the apoplast as co-receptors with membrane-localized TMK kinases. Auxin binding promotes ABP1-TMK interaction and activates the TMK intracellular kinase domain, relaying an extracellular perception into rapid intracellular phosphorylation cascades and expanding where and how cells can sense auxin. The surface signal is powerful and ultra-fast. ABP1's ABL-TMK activation drives a global phosphorylation response that modifies over 1,000 proteins within minutes of auxin exposure. This cascade targets H+ ATPases to control acidification and expansion, PN transporters to regulate polarity, biosynthetic enzymes like TAA1, and molecular motors such as myosin 11, producing immediate shifts in cellular machinery. A synthesis case study explains the classic paradox that auxin promotes shoot elongation, yet inhibits root growth. Both signaling branches contribute. But in shoots, the slow TIR1 AFB pathway and SOR mediated phosphatase inhibition sustain strong H plus efflux and elongation, whereas in roots, the rapid ABP1 TMK AFB1 CNGC14 module causes apoplast alkalinization and swift inhibition of elongation. A second case study shows how canalization and self-organization arise at the molecular level. The ABP1 ABL-TMK extracellular sensing module allows cells to read apoplastic auxin and selectively inhibit PIN endocytosis on the membrane facing higher auxin. That stabilizes PIN localization, reinforces directional auxin flux, and lets vascular strands self-organize through a positive feedback loop of transport and polarity. Bringing it together, the unified auxin network is a multi-layered blueprint where nuclear and extracellular perception, biosynthesis, directional transport, and phosphorylation cascades interact dynamically. The schematic emphasizes feedback on transport polarity, homeostatic cycles of synthesis and inactivation, and parallel intracellular and surface pathways that together generate context-dependent developmental outcomes. Finally, these revolutions open new frontiers and questions that remain unresolved. What are the direct molecular targets of second messengers like CAMP and CGMP? How does the global phosphorylation pattern translate into specific developmental programs? What mechanism lets cells sense auxin, flux directionality to polarize pins? And how do surface and intracellular pathways integrate to tune responses?